Here comes everybody.
All right. And Alex is unable to join as a panelist. Oh, there you are. Hey,
here.
welcome. It's so good to see you.
Good to see you too.
I was scrolling through like, what are our backup options? How's everybody?
Yeah, I just needed to update Zoom, I think.
Yep.
So,
It got me too.
Yep. Of course, the big day. And Alex, are you ready and prepared to present your own slides?
yeah, absolutely.
Okay, perfect. Awesome. I haven't gotten emails from anyone requesting that I present for you, so I'm assuming everyone is okay and ready. If at some point, no pressure right now, Alex, but if at some point you can upload those slides to the shared Google Drive folder. I just like to have them on record so that, you know, if, for example, anyone ever asks me for a letter of recommendation,
Okay.
then I
I
can.
mean, like, I can do that really quickly right now if you want me to.
Either way, no worries.
Okay.
Yeah, it can be later.
Okay.
Right. And then I'm just copying our schedule so that everybody can see it. All right. And okay. All righty. So it looks like we've got a load of people coming in. Awesome, awesome. And Michelle, we are live, we are recording. Thank you so much for helping get all this set up on the technical side of things. So with that, would love to welcome all of you. I'm going to share my screen for just a moment to have a couple welcome slides to get us started. First, a reminder of where you are. Thank you for joining us for this semester's Applied Ecology Minors Research Symposium. You are under no pressure or obligation to stay for the full 3 hours, but we will be here with awesome research every moment. So as a reminder, what you can expect is about a 10-minute presentation from each of the students who will be presenting today, after which we would love to open it up to open Q&A so that y'all can ask any follow-up questions that you have. We have the Q&A box you will see in your Zoom menu at the bottom. You can use that to ask questions. You can use the chat feature. Yeah. Any of the above. And then to our presenters, as you see questions come up, you can answer those if you like in text, via text afterward, if questions keep rolling in after our next presenter has begun. Okay, before we get started, I also wanted to take a moment to acknowledge that we at NC State University rests on the traditional territory of the Tuscarora, Lumbee, and Kawari tribes. The goal of applied ecology is broadly to study the natural world around us, which includes humans, right? And to apply that knowledge to address grand challenges. Often we can apply what we learn to better manage or conserve natural resources, but I think it's important to note that way before Western scientists decided that this was important to do and to study, expert knowledge of the natural world was already practiced and applied as an integral part of Indigenous cultures around the world. So while we are indeed very privileged today to celebrate undergraduate research, we need to remember that the Western, the scientific, the academic way is not the only way, and it is not even likely always the best way. An acknowledgement is a form of decolonization, which would be, steps toward dismantling an existing power structure. In order to change systems of oppression, we need to also acknowledge that the system is built on oppression. North Carolina State University is a land grant university, which means that it originally received funding through the Morrill Act. And the Morrill Act was federal legislation that bought or forcibly took land from Indigenous tribes. Indigenous land actually raised $134,920 to fund NC State. That would be the equivalent of $3,810,578 today. Just for some perspectives and reminders of our roots that are not always acknowledged. But pivoting from that back to what we are here for today, I want to return and thank you all again for coming, for attending our Applied Ecology Minors Research Symposium. And if you want to learn more about the minor, I will put our link in the chat. We have a webpage that you can find information about the program that each of our students has completed. That includes required coursework, as well as an independent research learning experience project, working with a research mentor either here at NC State or at any other academic institution, or with any entity beyond academia. So a lot of nonprofit, federal and state agencies, and as well as startups and business entities. So a wide variety of research mentors, of research systems that have been studied, of research methods will be showcased today. So thanks again. And finally, with each student, I will try to populate the chat without distracting from the presentations. This is our approximate schedule that we'll be following today. So Alex, you are up first. So I'm going to stop sharing my screen so you can start sharing yours. So thank you all again for coming.
Thank you, Dr. McKinney. So I'm good to just go ahead and start?
Yes.
Okay. Can everyone see everything okay?
That's perfect.
Okay. One second. All right, I guess I can just start like this. I just moved like all of the people to the bottom of my screen from the side. But anyways, thank you for the introduction. And so I'm gonna go ahead and start presenting. So hello, everyone. I'm Alexander Tung, and today I'm presenting a project that I've been working on that we'll call Trends in Research with Conservation and Human Infectious Disease Outcomes. So this was a systematic literature review project that I began working on in the Sustainable Health Ecology, or SHEL lab, since early spring of this year, so 2021. Okay, so the first thing I would like to discuss is planetary, one, and eco-health fields. Basically, they're like the same thing. They just go by different names depending on like which department or research institution is studying it. But the idea behind it is that it's the study of how the ecological connection that binds us all to all other organisms and ecosystems is so influential and interconnected in its function that the way that we treat the world's environment and ecosystems has a very direct effect on human health. So Currently, a lot of the focus in this field is on zoonotic disease. A lot of it is around monitoring, making projections about and analyzing current, past, or potential zoonotic spillover events. That's partly why the current COVID-19 pandemic is especially relevant to this field. Some of you may have heard of Eco Health Alliance a few times recently. They tried helping, I believe, like tracing origins with the pathogen, but I'm not an expert on that, so I'm not going to speak on that. But so we see a lot of research related to those kinds of things like zoonotic disease. But one thing is a lot of the literature tends to be oversaturated sometimes with things like review papers and perspective pieces. And those can be helpful and they certainly contribute to the field, but it's not as good for contributing to the growth of our understanding of planetary health as much as primary research will. So the project began with us deciding that we wanted to look for papers that hypothesized or measured at least one outcome for both human infectious disease and an outcome for conservation. So this is how it's kind of relevant to planetary health, is that intersection between conservation and human health. A good example of something like this would be a paper that discusses how vaccinating ecotourists in a lot of areas like natural lands that see a lot of ecotourism can help protect not only the humans themselves, but some of the animals like endangered gorillas. Like ecotourists getting vaccinated for COVID is quite possibly a very good way of protecting the gorillas from getting COVID as well. So the first step with this project is that you find the papers, because it's a systematic literature review. And to do that, we go through databases like Web of Science and like library search engines, and we use keywords like conservation, ecology, human infectious disease. So then we get our papers, and we got about 12,000, a little over 12,000 papers, which is not unusual for the beginning of a systematic literature review. You will get thousands of papers when you first compile everything. And a lot of that is because of how many keywords you use. We had around a few hundred. So also, like most systematic literature review projects, we're not interested in all 12,000 of those. And we're probably, it's unlikely that all of them are going to be relevant to what we're looking for. So that's where Colander comes in. Colander is a machine learning algorithm. And that did a lot of the sorting for us and basically tossing out papers that weren't optimal for what we were looking for. So long story short, we ended up with about 701 papers after that, and a further 69 were probably mostly sorted out by human means as we were evaluating the papers. So that leaves us with around 632 papers. So then you extract the data from the papers. So what you can see here is these are most of the categorical attributes that we evaluated the papers by. The only two things that are missing are I also evaluated things by funding information. That wasn't super related to the project, but we were curious to see where a lot of people were getting funding for a lot of this research. So the bulk of the data is on author information. It's on what their academic discipline is and like what kind of institution that they are doing their work for. So academia, it could be a research institution, public or private, a governmental organization, an international government organization, and so on. And then we have the paper science type right here. That's pretty important as well. That is essentially evaluating the paper on if it's primary research, if it's a review paper, just what kind of paper it is and what kind of primary research it is, if it is primary research. So that also includes things like theoretical papers and also other systematic reviews. And lastly, we looked at the geographic information. And the geographic information can be pretty self-explanatory, although just a note, it is about like what the study was, where it was about, and not where the study was done. So that's pretty important. So let's just get into the results. The first result is that the number of papers is increasing over time by a pretty drastic amount. That's not anything particularly special in terms of just like how the volume of publications works in most academic fields, although it is a pretty steep rise over time, especially towards the later 2000s. But I think the most significant thing is that there are no papers before 1990. And of course, that's a lot of that is because planetary health is a relatively new field, but not every paper that we looked at was strictly about planetary health. So I was very surprised by not seeing any papers from before that. So looking at the paper types, surprisingly, most of a very large chunk of the papers were primary research, which, if we recall back to what I said earlier about a lot of the papers in this field tend to be opinion or review papers. I think this partly has to do with the search criteria that we put into Colander when sorting out some papers. And because of the fact that we're looking for papers that are hypothesizing or measuring outcomes, it's more likely that it will toss things like perspective papers or review papers out and prefer primary research, although that's definitely not certain, because of course we still have about a fifth of the papers being review papers. But it is very interesting, like even primary cultural surveys on things just like local botany took up more space than perspective pieces. So moving on into the results with author information, not super surprisingly, most authors are academics. They're typically from either universities or they'll come from research institutions that are also a part of a university. But you also have a large amount of teams, because this graph is for all authors. So that would be an evaluation of the entire team that wrote whatever paper. And so those tend to be very mixed as well. So there's a lot of teams that have both academics and people that work for government organizations or non-governmental organizations as well. And then for my personal project, while doing this, I wanted to look at how interdisciplinary is the work in this field and things that are related to planetary health. And as I suspected, it's very interdisciplinary and by quite a large margin. So this is another look at all authors as a team for the papers. And we see a vast majority are mixed in their academic discipline. So basically what I saw when evaluating papers a lot was that it's mostly ecologists, environmental science, public health experts, people in veterinary medicine. There was a lot of those. People in geography, cartography, conservation, and even green and civil engineering. So geographically, it has a very, very broad and global focus. You can see here with the pie chart just how many countries we're dealing with. And then we have a share of paper focused by continent as well. If you see the NA there, that is accounting for papers that either that they didn't have a specific location in mind, or it's really just a commentary on just broad or global phenomena. So that's like a lot of review papers and public health papers, and also a lot of perspective papers. So in conclusion, we confirmed some things that we expected for sure. Most importantly, we confirmed that planetary health is an increasingly interdisciplinary field in terms of the work that's done within it. It's highly collaborative as well across institutional lines. And we were also surprised by some things too, something we were surprised by was that ecological papers tend to measure and hypothesize outcomes for human infectious diseases, as well as obviously ecological phenomena. And they were more likely to do that than things like public health papers were to evaluate outcomes for conservation or the environment. So that was definitely something we were surprised by, and that can kind of say something about the way that both of those fields work in terms of how they're viewing subjectively their studies. And finally, I think it's pretty nice that we now have this very large database of papers that are sorted by all this criteria. I think that will be very helpful for future research on planetary health. And that is it. If anyone has any questions, I'd be happy to answer.
Awesome. Thank you so much for getting us off to such a strong start this afternoon, Alex. At this point, I will reiterate what Michelle has shared in the chat. If you're feeling bold, feel free to use the raise your hand function, and I will unmute you so that you can ask your question aloud. Otherwise, we've got the chat box or the Q&A box. If you have any questions, please share them at this time. All right, we have our first question from Alex Nelson in the Q&A. Great job. Do you remember if there were a lot of interdisciplinary COVID papers, or do those mostly come from the medical field?
Sorry, the button for like muting and unmuting is being really weird right now, but can you repeat that question one more time just so I have a good...
Sure. Do you remember if there were a lot of interdisciplinary COVID papers, or do those mostly come from the medical field?
So I know that we're gonna like want to talk about COVID when related to this, but unfortunately, most of the papers didn't quite make it to 2021. We only had about one paper from 2020. I'm not sure why we only had one paper from 2020, but we also had like none from 2019 and 2021 either. But there was definitely a lot of papers. I evaluated a lot. I evaluated like over 425 papers. And so I came across a lot of papers that are very similar to what you would see with in terms of like trying to analyze the events over the course of a zoonotic spillover event. And yes, those are usually very interdisciplinary. There's, it's usually strongly dominated with people in veterinary medicine and public health. And then you'll usually have maybe an ecologist in there as well. So I would say when you start to see more papers on COVID, you could certainly see that. Yeah.
Awesome. Thank you so much, Alex. And to Alex Nelson for your question. In the interest of time, I'm going to ask Alex Tung to go ahead and stop sharing your screen so that we can welcome Eileen to the virtual stage. Thank you again for kicking us off.
Oh, here we go.
So Eileen, if you'd like to share your screen.
Okay, let's see, get this organized. Oh, hold on. Let me fix that real quick. I don't like what I was doing. Oh, whatever. Okay, cool. All righty. That seems good. All right. Good afternoon, everyone. Can everyone hear me and whatnot?
Yep, sounds
Okay.
great.
So my research is called The Freshwater Snare That Lasts Hundreds of Years, a case study of derelict fishing line accumulation along the shoreline of Jordan Lake, North Carolina. So first of all, I started looking at plastic pollution in general, and there is way more plastic pollution research in the marine environment than there is in the freshwater environment. And that trend follows along the same idea when you're looking at fishing line. And in the marine environment, it's much more heavily focused on the fishing industry as opposed to recreational anglers. So it was super hard to find any research done on freshwater derelict fishing line. I only found like 2 good papers, but this is a picture that I took while doing my research. And you can just see what a mess some of the fishing line can look like. So back in the day, before plastic was invented, fishing line used to be made out of biodegradable materials such as hemp and cotton. And today, the vast majority of fishing line is made of plastic. And this plastic can persist in the environment for hundreds of years. And this is another picture that I took. And it seems like fishing line attracts more fishing line. So it just kind of bundles up into this huge rat's nest that can be super fun to deal with. But this line, some of it would just crumble apart as I would measure it. And so it's kind of hard to tell how long some of the line has been in the environment because it can persist for so long. So why does this matter? This matters because it creates an essential, inescapable snare for wildlife. And fishing line and derelict fishing gear can continue to do what they call ghost fishing long after the line has been discarded or lost. So this little guy that we found, it looks like someone was fishing, caught him, and when the water was a lot higher, he got caught up in these little thorny weeds, and the person fishing cut the fish loose, and then the fish was unable to escape and he died. He or she, I'm not sure what the gender of the fish was, but it can persist in the environment for a very long time. And this can be the fate of a lot of animals if people don't find them to help them escape. So my case study area was at Jordan Lake. It is a very, very popular fishing lake in North Carolina, and they do bass fishing tournaments there all the time. It has 180 miles of shoreline, so it leaves a lot of area for people to fish. And last year, or in 2016, 30.1 million Americans fished in freshwater. And freshwater fishing is the most popular type of fishing in the United States. When you think about that, it makes a lot of sense because people in like the central part of the United States can't really get to the ocean to fish. So we have a lot of research done on the marine environment, but in the United States, a lot of fishing is taking place in the freshwater environment. So the questions that I came up with and my hypothesis. So I wondered, how much derelict fishing line is there to begin with along the shoreline of Jordan Lake? And are certain amenities that are provided at these fishing locations contributing to higher or lower rates or accumulation of derelict fishing line? So my hypothesis was certain amenities will contribute to a higher accumulation of derelict fishing line. So this is one picture of after we finish collecting at a spot, and that's a 5-gallon bucket that's full. So the variables that we took into consideration at each site that we looked at were trash cans, fishing piers, park entrance fees, parking lots, bathrooms, boat ramps, campgrounds, parks, and hiking trails. So here's another picture that we took. And as you can see, there's no trash cans provided at this fishing pier. And the fishing pier has a very large parking lot and it's super easy to access, but there's no trash cans provided. And so as you can see, some people are not adhering to the request to take their trash with them. So the site selection methodology, we wanted to keep it as random as possible because my husband and I love to fish at this lake, and we did not want to have any bias when selecting locations because there were some sites that we went to that had a lot of fishing line, and we wanted to make sure we were being as random as possible. So we selected 40 sites using Google Earth images. So essentially we zoomed in and looked for 40 locations that appeared to be accessible to fish. From those 40 sites, we narrowed them down into 20 sites, 10 and 10 being high traffic and low traffic. And then we used a random number generator to select 20 sites, and 10 being low traffic and 10 being high traffic. But we didn't use that when we were really looking at the locations. We just used it for our site selection for the high traffic and low traffic. Now for the line collection methods, it was all visual search methods. And the average amount of time that we spent looking for fishing line at each site was 65.33 minutes. But the least amount of time was 28 minutes and the most was 123 minutes. And the dates that we collected was the 15th of December through the 8th of May, 2021. And the reason that the timing was different is because each site, we essentially checked each site until there was no more line to find. So some spots had very little line and some spots had a lot of line. So we wanted to make sure that we collected as much as we could. Even if it took us longer to do, we wanted to make sure we cleared the site of all the line. So here's a really cool interactive map that we made. And so we have 20 sites that I've collected line from, but only 18 sites have been measured so far. So these yellow dots represent the line that I haven't measured yet. But each one of these spots you can click on and it'll tell you the location of the line, the location of the site, the amount of line we collected in inches and in meters, lat and long, number of pieces, how much time we spent, and then all the amenities that are also provided at that location. And you can zoom in on this map and it gets super cool, especially because I want everyone to remember how flooded this is, because I'll bring that up later. So these are each one of our little sites. So for the items, the amenities that we looked at, we noted them in an Excel spreadsheet with the amount of the meters of line, and then we turned it into a binary code. So the boat ramps, like anything that has a yes equals a 1, anything that is a no equals a 0. So the measuring methods, each piece of line we had to separate from any attached fishing tackle and any debris. And then I untangled it, measured it in inches, logged it, and then we calculated it to meters in the Excel spreadsheet. And this is all fishing line in here, all of it. And so we had to pick up that big ball and then clean it and yeah, measure it. So my results are actually pretty cool. We did a stepwise regression through Excel, And the two statistically significant variables came up as parking lots and fishing piers. So essentially anything that gives accessibility for fishing, that's like the easiest access for fishing. It's like a designated provided location for fishing was the most statistically significant. And here's just a really cool heat map that we decided to make too, just to be cool, but you can see this area is pretty intense. And I think this area will be pretty intense too when I finish my last locations of measuring. And here's the length of line measured. So you can see like how small, but every single site had fishing line. Every single site had some amount of fishing line, whether great or small. And here's the percentages that we found too. So the Beaver Creek Fishing Pier was 15.5%, and the fishing access at Beaver Creek is 18.3%, which is interesting because there's no real access there. It's kind of like you park on the side of the road and then you hike down to fish. So here's all of our total measurements taken. Like I said, 18 of the 20 sites have been measured so far. And the total line so far is 5,513.88 meters, which comes out to 3.426 miles of fishing line. And here's all the other items that we've found so far, which is pretty cool. The total sinks, those are lead, and those are lead fishing sinks. We found 128 of them so far. So for the discussion, what do these results mean? Well, based on the p-values, it means that there is a probability of a connection between amenities provided and the amount of derelict fishing line that accumulates at each site. But there's a multitude of other factors that we need to take into consideration, such as human behavior and how fishing line is actually lost. If fishing line is lost on accident, like this picture that I have here, it looks like someone was fishing, they overestimated how much power they needed and lost their fishing line to a tree. And so that's going to be there for a very long time. Or if it's on purpose, or if it's just absent-mindedness that creates so much derelict fishing line. Also, the water level change, if you remember from that picture, you could see how high up the water levels can get. So that moves a lot of fishing line around. It'll take fishing line from a certain shore and bring it to another spot. And then wind as well, because the lake doesn't really have currents, but there's a lot of wind that moves the water around on the lake. So you have to take all those things into consideration as well. And then the fact that there's been very little research done on this topic, and I'll get into why that might be. So our caveats, the research is tedious and time consuming. If you think that you have patience, it's probably a good idea to unravel 3.5 miles of fishing line and see how that goes. It's definitely given me a lot of patience. The lake level was a significant contributing factor to when we could take samples. So that's why it took so long to be able to collect samples, because if the water level was up 15 feet, I could not go out and take samples from a fishing location because it'd be underwater. And we really had no way of knowing how long the line was there. When you look at this picture, some of this stuff is how we were finding line. It was buried underneath a lot of sediment. So we don't know how much line is underneath the sediment as well. So we attempted to create a logical timeline of when to go collect locations. And I thought I gave myself plenty of time, but I've only finished measuring 18 of the 20 sites. Like I said, we checked the water level, and this could be a continuation study easily. Of clearing a whole site location and measuring it over time to see the actual accumulation rates from a clean slate. And so this is just a cool picture that I took. I wanted to share. This is all line that's buried. And this was not uncommon to see. Like, we saw a lot of this, and a lot of line we couldn't collect because it was buried so deep under sediment. But other than that, I'd like to say thank you to my research mentor, Dr. Leggett. She has been awesome. She's been helping me with this research for over a year now. And this is my research crew, my husband and my dog. And this is one of our last days out on site. And then here's some animals that we came across while we were out doing our research. But other than that, I'll stop sharing. And if anybody has any questions, I'd be happy to answer them.
Thanks so much, Eileen. I had a question that I dropped in the chat for you, just to get us started while other folks collect their thoughts and their fingers for typing. So that Beaver Creek trend with actually more debris at the access point was really interesting. I wonder, is it downstream from the pier? Like, do you think it's a proximity thing or?
It's a very, very popular fishing site. It's a fishing site where people spend a lot of time. So people will sit there all day sometimes and fish. So when we found a lot of the line at that spot, it would be like near a tree stump that was sitting. So you could tell people are like sitting and chilling there. And then there'd be like balls of fishing line there. And then that's the spot that we found a lot in the sediment as well. There was a lot of fishing line buried beneath the ground, which was crazy.
Wow.
Yeah.
Thanks. And in the chat, Shannon Dolan has asked, why did you select a story map to present your research?
So I took GIS in the spring and I thought it'd be really cool because I do want to share this information with the parks after I'm done measuring everything. So they might be able to use this if they do decide to put amenities such as trash cans, or they have these really awesome fishing line recycle tubes that maybe they could put up and see if it helps to lessen how much derelict fishing line is there.
And Dr. Elsa Youngstedt has asked, has said, great talk, and hope I didn't miss it, but what actions do you recommend should be taken to reduce or manage derelict line?
And that's a hard part, is because it's hard to figure out what's really making this happen. And a lot of it might be absent-mindedness or getting hung up. Like, I know I fish a lot, and when you get hung up, you can't really do anything about it, you know, unless you have waders on. And I've done that. I've put on my waders and I've gone in after my line. But in a lake like Jordan Lake, that can be difficult to do. I think it comes down to like education, mostly just making people aware, because I don't think people are maliciously doing these things. I think it's just something that's absent-minded. So education and providing the amenities that people need. Like, if you're going to provide a fishing pier, like that picture I saw, it's going to be a very popular spot to go and spend a lot of time. So it might be worth it to provide the amenities as well of fishing line, recycle receptacles, signs, and education amongst the fishing community, because it seems like communities can influence so much more than just some random person. You know, like, I've been out fishing and I lost my mind when I saw one of my friends throw their fishing line on the ground, and guess what? That person hasn't done it since then. So there's a lot of community that can support and build that.
Awesome. And we got a twofer, because you have answered Corrine Morales' question. Do you think the public outreach and education would help? Often through peer education. Right. And then one final question. Trey Jeffers has asked, do you think that fish and other wildlife species that burrow into the sediment of lakes could get caught in the fishing line buried in the sediments?
Absolutely. And there's this really interesting paper that I did read, and it was talking about plastic pollution, how it's affecting like worms and other animals that live in the soil. And that's actually what I'm going to focus my research on for my master's degree, is looking at plastic pollution in the soil and in the water. So not just what we can see, but what's going on that we're not seeing, because plastic does leach its chemicals and it does create a lot of issues for that as well. So it could be very possible a vole could be tunneling through and get caught up, and we would never know about it. And there was a lot in the sediment so my measurements aren't even covering everything. It's just covering what I could get to. So it's crazy to think about.
Lots of layers.
Many layers, yeah. And there were trees that fell over, And in their roots would be tons of fishing line. It was just decorated, tons of fishing line. And we would climb out and be crawling on these tree roots, trying to get out everything we could. And that's another reason we focus mostly in the winter time, because like I said, I found that lovely copperhead, and that was in May. So we're digging around a lot. So we tried to do it in the winter so we wouldn't, be disturbing any wildlife or, get tagged by a snake who's defending her home. So
Awesome. Well, thanks again so much, Eileen. Up next, we have Shelby Williford. So feel free to share your screen.
Can everyone see this and hear me?
Yes, perfect.
Awesome. So my name is Shelby Williford, and for the past few months, I've been working on this project studying how turkey litter can be used as a fertilizer for industrial hemp. So first off, what is hemp? Cannabis sativa is one of the world's oldest cultivated plants. Sativa actually means cultivated and is generally used for plants that have positive health impacts. Now, I know a question a lot of you are probably thinking is, what is the difference between hemp and marijuana? They are two different cultivars of cannabis. You cannot tell them apart just by their physical appearance, but they have drastically different chemical compositions. The two main chemicals inside of them are THC and CBD. THC is a psychoactive chemical that can cause intoxicating effects, while CBD is not psychoactive and has been linked to some positive health benefits. So hemp legally cannot have more than a 0.3% concentration of THC, but tends to have higher levels of CBD. Marijuana, on the other hand, has an average of 10% THC and less CBD. For many years, both these plants were viewed the same in the eyes of the law and were both illegal under the Controlled Substance Act of 1970. However, the U.S. Agricultural Act of 2014 permitted pilot programs for industrial hemp research. Then in 2016, the North Carolina General Assembly legalized production of industrial hemp and left a lot of the hemp research in the hands of the land-grant universities, so here at NC State and at NC A&T. So one of the reasons that hemp is such a valuable crop is that it is extremely versatile. One of the big markets for hemp is its use as a fiber. It is sustainable, high yielding, and is one of the most durable forms of natural fibers. Hemp is composed of around 20 to 30% long outer fibers and 70 to 80% of short inner fibers. The longer fibers tend to be more valuable, but both of them have their own purposes. Some of the things they can be used for are textiles, paper. They also have the potential as a biofuel crop. And like Eileen just said, they can be used for fishing lines. Another market for hemp is as an oil or hemp seed. So hemp seed has been used as food for 3,000 years because it is rich in essential fatty acids. Can also be used for things such as like oil, painting, etc. One of the largest markets for hemp is as a pharmaceutical due to its high CBD concentration. As I mentioned earlier, CBD is not psychoactive, so it has no intoxicating effects, but it does have potential to reduce things like inflammation and anxiety. Sales in the pharmaceutical are expected to lead all hemp-based sales by 2022. So to yield the best results, many producers use fertilizers on all types of crops. One method of fertilization is the use of manure as a fertilizer. Animal manure has been used for generations because it can leave positive impacts on soil health. When used correctly, manure fertilizers can be more sustainable than synthetic fertilizers. Some common types of manures come from horses, cows, and chicken. There's a huge potential to incorporate manure into hemp production, but so far little has been reported. So this gets us into the objective of our study. The overall objective is to compare the use of turkey litter as a fertilizer versus a store-bought synthetic fertilizer. In the study, we used the brand Dr. Earth. Another thing we wanted to look at was how different soil types affected the growth of hemp. So we collected soil from NC State's Chink Forest and the hillside in Durham. We also used potting soil. Lastly, we wanted to see how different genetic makeup affected the growth of the plants. So we use both male and female strains of Electra and Spectrum hemp. So to set up this project, we collected 50 gallons of soil from both the Chink Forest and the hillside, and then homogenized it so it was all mixed up and good to go. Then we made sure to plant an even number of the hemp with a certain gender and strain in each type of potting soil. We had 99 plants in total, and the plants came in about 3 waves just because it took time to grow and develop them to the stage they needed to be in to be planted. And there's usually about 1 or 2 weeks between the different waves, so they were all about at the same point. Once all of the plants were planted in 1-gallon containers, we started fertilizing them once a week. Half the plants got 1 teaspoon of turkey litter, and the other half got 3/4 of a teaspoon of Dr. Earth. Then for the next few weeks, well, months actually, the plants were measured and watered 4 times a week until the end of the study, when we collected the final measurements and we measured the diameter of the stems. We also collected the above ground shoots of the plants and dried them and weighed them to get the final biomass. Unfortunately, we were not able to save the roots to also measure the below ground biomass as well. So I hope this GIF is playing on your guys' screens, but one of Dr. Leggett's grad students, Tyreek, gave me the idea of taking the same picture at the end of the table every time I went to measure the plants. So then at the end, I could put the pictures together to kind of show a time-lapse progression of their growth. I'm not sure how well you guys can see it, but some of the plants in the back over here grew to be very large, which was really cool to see. We also had fans in the greenhouse to keep the plants cool, since anyone that has ever worked in the greenhouse knows how hot it gets in there. For the last few weeks of the project, we also transplanted some of the larger plants into larger containers just to make sure that their growth was not being hindered by the 1-gallon containers. So this was also done by Tyreek, since I had to leave for a summer internship. But so he transplanted some of the larger hemp plants from the 1-gallon containers into 5-gallon containers. For our results, I have the average diameter and height for the hemp plants broken down by their fertilizer treatment. Unfortunately, we had higher than anticipated mortality rates, so we could not compare the effects of the soil types or the genetics of the plants. Overall, for both height and diameter, Dr. Earth showed higher growth than the turkey litter. So this chart has the biomass for all the plants, again, broken up by fertilizer treatment. And even here, Dr. Earth still had overall larger numbers than the turkey litter. However, we ran a significance test with a p-value of 0.05, and we found that there was no statistical significance in the difference between the sizes of the 2 plants. And that was pretty exciting to see that turkey litter does have potential as a fertilizer. So as I mentioned earlier, we had high mortality rates within this project, which ended up being a challenge at the end because that greatly limited the tests and comparisons we can make. As I said, we started out with 99 plants and we only had 24 make it to the end. So if this project was replicated, it'd be great to have a larger sampling size at the end, so then we could compare the effects of the soil or the genetics. But the overall takeaway is that turkey litter does have potential as a fertilizer for hemp production. So while it did show slightly lower growth averages than the synthetic fertilizer, the results were not statistically different. It was significantly statistically different. So we think that it does have great potential. And some applications of this is that it would just be a more sustainable fertilizer option. And it would also be great for producers that also raise livestock. So then they could take the manure from their own fields and turn it into fertilizer. So I'd like to give a huge thank you to Dr. Zakia Leggett for allowing me to work on this project with her. I'd also like to thank Dr. Michael Harrington and Dr. Theo Lehman for supplying the hemp plants. And lastly, I'd like to thank Tyreek Cooper for all the help that he provided in this project. And that is it. So thank you for listening. I'm happy to answer any questions.
Thanks so much, Shelby. Has anybody got questions? Feel free to put them in the chat, or you can raise your hand and I will unmute you, or you can put them into the Q&A. If you think,
I have two questions about the high mortality rate. So we know one issue was when I had to leave for my summer internship, I left like instructions on how I'd been taking care of the plants, and whoever came after me to take care of the plants, there was some misunderstanding on how much they need to be like watered and taken care of. So that was definitely a big impact on that. I don't think there's too much of a differences in loss between the two treatments. I think at the end of the 24 plants left, it was pretty evenly split between the plants I were treated with, Dr. Earth, and the turkey litter.
thanks again, Shelby. Up next, we have Claire Waters presenting. So Claire, if you would like to share your screen.
Yep, gotcha. Alrighty, everybody. Let me just sit. Yeah, so my name is Claire Waters, and I'm here today to talk about the impact of an endangered ecosystem engineer on songbirds. And I did this project with Dr. Michael Cove at the Museum of Natural Sciences. So just to give you all a brief overview of everything I'm going to be talking about today, we're going to start off with an introduction to the key largo wood rat, which is the endangered ecosystem engineer in question. Then we're going to go over my hypothesis and objectives, methods, results, and then we'll get into a discussion of everything. The key largo wood rat is an endangered subspecies of the eastern wood rat, which is endemic to Key Largo, Florida, as the name suggests. Its populations are threatened primarily due to urbanization, due to loss of habitat from development, and also invasive predators, mainly house cats. And so since they were declared as an endangered species in 1984, the U.S. Fish and Wildlife Service has begun a restoration project to build stick nests to provide habitat for the species, trying to emulate stick nests like the one shown here. So under normal circumstances, wood rats build stick nests, thus their status as an ecosystem engineer. And these stick nests have unique microclimates, which consist of moderate temperatures and high humidity relative to the surrounding environment, making them an important habitat for many species. These stick nests are also associated with areas that are high in soil organic matter. And due to these two factors, they have also been found to have an increase in arthropod abundance and diversity in the stick nest and in surrounding areas. And so it has also been found that insectivorous birds can be used as an indicator of arthropod populations because they are directly consuming these arthropods. And we also know that birds can be used as an indicator species to assess ecosystem health at large. But despite the fact that we know that there are these increases in arthropod abundance and diversity, we don't know how that is affecting bird communities surrounding stick nests. So because of that, the objectives of this project were really just to investigate the bird communities surrounding wood rat stick nests in Key Largo, Florida. And we hypothesized that we would find a higher biodiversity of birds in the presence of the Key Largo wood rat. But we also acknowledge that cats, predators, and development might also have an effect on bird species richness. So our study site was Key Largo, Florida. It included the Crocodile Lake National Wildlife Refuge as well. And this map here just sort of shows the distribution of all of the birds that we ended up observing throughout the study. And each little dot represents an individual with the key over here to the right. Unfortunately, I don't have like a experimental design or anything to share with you guys because we actually retroactively analyzed data for this study. So in late 2018, camera traps were set up throughout Key Largo, Florida, with the intention of studying wood rats. And originally, only mammals captured by this camera trap deployment were identified to species. So when I started doing this research with Dr. Cove, he helped me to access eMammal, and then I went through and identified all visible birds to species. And for bird ID, I also used things like eBird, Florida Bird Guides, and occasionally Seek for aid in identification. So now to get into the statistical analyses of this project, our sample size consisted of 730 sites. And once we had collected data on all these bird species, we really wanted to analyze bird species richness with covariates such as, the abundance of other species and the presence absence of these species as well. So more specifically, we ended up looking at bird species richness with covariates of abundance and presence absence for the following species: the Key Largo wood rat, house cats, Virginia opossums, and northern raccoons as well. And then we also looked at the combined effect of all of the aforementioned predatory species. And then we also looked at some abiotic factors like distance to development, distance to forest edge, and also the presence or absence of stick nests at the site. So we did all of this statistical analysis in RStudio. We ran generalized linear models and with Poisson distribution. And yeah, we sort of did all of this to retrofit align with our data and get a slope to sort of gain a proxy for the relationship between these different variables. But importantly, we acknowledge that these aren't the only variables that might affect bird species richness. This was just the data that we had. So I'm going to get into some of the results with the analysis that we did now. So some summary statistics to start off. As a part of this project, I identified 306 birds. I have shown the stats for the 6 most abundant birds here on this slide, but I also identified warblers, thrushes, some doves and hawks, a willet, a grackle, but by far, the ovenbird, which is this bird pictured here, was the most abundant that was captured by our camera traps. And importantly, so note, there was also Key Largo wood rats at 77 sites, stick nests at 73 sites, and cats at 43. So the results of our statistical analyses, we found a statistically significant relatively strong positive relationship between wood rat presence absence and bird species richness. So as you can see on this graph here, the blue line represents bird species richness when wood rats are present, and the red line represents bird species richness or predicted bird species richness when wood rats are absent. So that supports our hypothesis that we were expecting to see that increase in bird species richness with the presence of wood rats. However, we did also find a really strong negative relationship that was also statistically significant between the presence of cats and bird species richness. So as you can see, when cats were present, it's just essentially predicted that there will be no birds at that site. And we also did find a slight positive relationship between distance to development and bird species richness, meaning that as distance to development increases, as we get farther away from development, we're seeing more birds. And that's what's on the x-axis for both of these graphs. So you can see that with the presence of wood rats, absence of wood rats, and in the absence of cats, we're seeing that increase in bird species richness as we get farther away from development. And we also ran a test actually this week on wood rat stick nests and bird species richness, and we found an even stronger relationship than the relationship shown here between the presence of stick nests and the diversity of birds. And it's important to note that these models that I've discussed were the best models that we ran based on AEC values, with the exception of one model that we ran with possums as the covariate, possum abundance as the covariate. Yeah. So now to just kind of wrap this all up, we did see that positive relationship between wood rats and bird species richness that we were expecting to see. And we can surmise that this was probably due to the fact that there's an increase in arthropods in the stick nests that wood rats are building. So let's see, sorry, I lost track of my words there. So we can sort of guess that, or this research rather shows that wood rats may have cascading effects on the ecosystem at large. If their stick nests are fostering high diversity of insects and that's fostering a higher diversity of birds, then that could have cascading effects on the ecosystem. However, we did see that really strong negative effect of cats on birds, and this is actually a dual effect because the cats also pose a serious threat to wood rats as well. And so what this means is that the cats may be directly killing birds, but then they're also dampening the positive effect that wood rats have on bird species richness because they are also likely directly killing the wood rats as well. And so based off of this research, we know that wood rat conservation has implications for other species, especially now insectivorous songbirds, such as the American redstart shown here. So I hope that this can guide future policies and management. And yeah, I also hope that research in the future might analyze other covariates that we were unable to look at in this study, just to get a better picture of these bird communities surrounding wood rats. And so I want to give a special thank you to my research mentor, Dr. Michael Cove. He was very, very helpful during this project, and I couldn't have done it without him. So thanks. And yeah, these are my sources. Any questions?
Thanks so much, Claire. I am opening up the Q&A in the chat to make sure we don't miss anything coming in.
Thank you.
You've got a question in the Q&A box from Madison Smith. You mentioned that you hope to enact policy changes as a consequence of this project, but do you have any other broad goals for this study or larger implications on the surrounding ecosystems?
I definitely think that there's probably further research needed in this area. As I said, I think that the results of this study show that wood rats could be having cascading effects on the ecosystem, but we would need further research to determine their effect on, say, other mammals or maybe predatory bird species, stuff like that. But I do think that this shows, since we saw that positive relationship between wood rat stick nests and bird species richness, that the project that the U.S. Fish and Wildlife Service is doing is really important because those stick nests are important to birds. So it reinforces the importance of that project specifically. And I see another question from Jim Rice in the Q&A. If I recall correctly, you had stick nests at almost all of your camera sites. Do you have comparable data on sites without stick nests for comparison? So we actually had 730 sites, and there were stick nests at only 73 of those sites. So yeah, it's a little bit confusing because we had 730 sites total. There were wood rats at 77 of those sites, and then stick nests at 73. So the reason that we also analyzed stick nests and bird species richness as opposed to just wood rat presence absence in bird species richness was because wood rats are present at sites where there are not necessarily stick nests. But yeah, to answer your question, there are sites, a lot of sites that don't necessarily have stick nests at them.
And Jim in the chat said, thank you for clarifying that. And nice study. Well, thank you very much, Claire. If you can stop sharing your screen, perfect. Then next we will welcome Andrea Padilla Guerrero to the virtual stage.
Hello. Let's see. Can y'all see that all right?
Yes, perfect. We can hear you well too.
Okay, perfect. Well, my name is Andrea. I did a research over the summer and it was focused on investigating controls in the production versus diffusion of carbon dioxide and methane at soils at 3 different topographic positions. All right, let me see, there you go. And so my research was focused on tropical forest, and they are some of the most important biomes to understand the impacts of climate change. And they play a key role on the global carbon budget currently. And so with climate change, there could be effects to the biome's ability to store carbon. And with the changes that we're seeing in climate change, it could affect the amount of carbon dioxide and methane emissions from the biome, thus exacerbating the greenhouse effect. And in this image, we can see that carbon dioxide and methane are some of like the greatest greenhouse gas emissions from the US. And so that's why I decided to focus my research on those 2 greenhouse gases. And so I focused on researching topographic location, biotic variability along the depth profile, and other influencing factors in soils, because those are the ones that affect the concentration and diffusion of CO2 and CH4 in the atmosphere. And so other factors are also soil temperature, moisture, and oxygen levels, and all of those impact the biogeochemistry of the soil itself. And so the objectives of my research were to determine the relationships of those abiotic components that I just mentioned, and see what the relationship was to the production of both CO2 and CH4 at 3 different depths along the soil profile, and see how that affects the production or diffusion of carbon dioxide and methane in the tropical soils that I researched. And it was also to record the concentrations within those soils and just look at the overall relationships between the production and the concentrations as well. And so one of my hypotheses before the research started was that the higher the concentrations of moistures in the soils were, basically that would mean that there would be lower rates of oxygen in those soils, causing a higher concentration of methane because of those, because of the lack of CO2 in the soil itself. And so the place that I did my study was at the Tropical Response to Altered Climate Experiment location in the eastern part of Puerto Rico in the Luquillo Experimental Forest. And on the bottom right corner, we can see the Savannah Field Research Station where I did my research. And so, like I mentioned before, I focused on observing temperature, moisture, and oxygen, and their relationship between the production of carbon dioxide and methane at 3 different soil depths and topographic location. So my 3 soil depths were 15 centimeters, 40 centimeters, and 100 centimeters. And those were at each of the topographic locations, which is near a ridge, mid slope, and lower slope. And then we took all of that and also took flux measurements of the gases themselves. And so some of the instruments that I used for my research were soil pits, which I used to characterize the soil depth profile in terms of its microbial function. And I also used moisture, temperature, oxygen sensors, and soil equilibration chambers, and as well as an LI 7810, which I will show in this picture here, and an LI 800A, which helped me take air samples from the soil pits to be able to record both the concentration of the gases as well as the flux of those gases. And so here we can see the temperature of the pits. And of course, you know, we can see that at the 15 centimeter mark, which is the orange bars, we can see that the temperature was the highest. And as the depth goes deeper, that temperature drops. And in my topographic location number 3, we can see that those temperatures are a bit skewed. And so I faced the challenge that my sensors were broken in pit 3. And so same with the water content in the soil. So at the surface levels of the pits at 15 centimeters, we saw the lowest due to evaporation. And then as we went deeper into the depth profile of the soil, we see that there is higher concentrations of water. And then same with oxygen, we see that those levels are higher at the surface, or lower at the surface and higher along the depth profile. And then again, with all 3 of those measurements, my topographic position at number 3 was unfortunately, like the sensors were unfortunately broken. So those measurements were a bit off. And so with methane concentration, we can see that we have our control, which is the yellow bar. And so methane in the atmosphere is higher than it was in the soil. So that means that there is net methane consumption. And so that means that more methane is being consumed than is being produced. Let's see. And then with the CO2 concentrations, the opposite actually happened than CH4. And so atmospheric CO2, which is the yellow bar, is lower than the concentration of CO2 in the soils. And so that suggests that the CO2 being produced is most likely having a hard time being diffused. And so there may be limitations, and some of those limitations may be the water content in the soils, which may be filling up the pores of the soil itself, which therefore does not allow the oxygen to diffuse into the atmosphere. And so here we can see that the methane flux, meaning like the methane that was released from the soil, well, it's actually negative. So nothing was released from the soil itself. And that is because the methane was higher in the atmosphere, meaning that there's no diffusion and there is overall net methane consumption. let's see. And for the CO2 flux, these are pretty small values, but it is, like I mentioned, lower because of the limitations on the diffusion, which is, like I said, a possible cause of the water infiltration in the soils themselves. And so some next steps would be to continue my research. I didn't have very much time to conduct the research itself. I was only in Puerto Rico for about a month. And so that included just getting settled into my research area and also learning how to use the machines. And so I only had a few days to actually collect samples. And so I think for this research to really be able to represent the results, I would need to go back and have longer time to collect data. Let's see, and so my acknowledgements go to the NSF program for funding this research, to Alonso Ramirez at NC State University for allowing me to join them in the program, as well as Trace. Can y'all still see my screen?
Yes.
Okay. And it is the slide?
Yes.
Okay. And so I would also like to thank Alonso Ramirez for allowing me to join the program, and Dr. Tiana Wood, who is my mentor or was my mentor for the summer, in helping guiding me through this research. And I hope to be able to, at some point continue it in the future. Let's see. And that is all.
Thank you so much, Andrea. You see, you have a question in the Q&A box from Jim Rice.
Yeah, so do you have any ideas as to how the patterns you saw might be affected by different surface vegetation cover? So I would say that based on, I didn't look at this specifically, but I would say possibly the root biomass in the soil might affect water concentrations, oxygen availability. So I don't have a direct answer as to how they were affected based on like the vegetation covers around the top topographic positions. But I do think that root biomass and CO2 concentrations would be affected by vegetation cover.
Thank you.
Of course. And I will stop sharing my screen.
All right. Thanks again. And up next, we have Adrian Chamberlin.
All righty. Okay, can you see me? Can you hear me? Can you see my screen?
Yes, all systems are go.
Excellent, thank you very much. So, hello everyone, my name is Adrian Chamberlin. I'm a 5th year senior studying fisheries, wildlife, and conservation biology. And this past semester, I have been working to conduct a historic mammal inventory of a local urban wetland. So why even have an interest in urban wetlands? I particularly am interested in them because they are unique in that, as the name implies, they are surrounded by metropolitan and city development and are often fairly isolated. They don't usually have a lot of contiguous green space, so they're kind of existing in their own little bubble ecosystem. But they perform all the same functions that any other wetland would. They filter rainwater before it returns to any primary body of water. They provide habitat for wetland plant and animal species. And particularly urban wetlands allow for city residents to have green space that they might not otherwise have access to, which is especially important in childhood education and fostering interest in nature. And it is extremely important in mitigating flooding in developed areas. Raleigh is a very good example of this, especially given that our metropolitan area is rapidly expanding. And we're seeing lots of increases in more non-porous surfaces. This picture on the left is on the Triangle Greenway on the Walnut Creek Trail when it flooded in May of 2018 after a particularly heavy rainstorm. So this isn't even after a hurricane. This is just a lot of rainwater. So this is when Walnut Creek flooded, and that is the particular watershed that we're looking at today. The Walnut Creek watershed services much of Southeast Raleigh, or South Raleigh in general, and then Southeast Cary. Walnut Creek is there outlined in light blue. So it's very important that the city is maintaining the natural wetland areas, the riparian areas they have, and mitigating drainage around those, because there is a whole lot of suburban development, especially occurring around these areas. And then as you get into southeastern Raleigh, a lot of industrial development. This is a little bit of a side note, but the city is implementing some more artificial wetlands along a lot of near the Walnut Creek areas, but that's not the focus of today. Just a little interesting note, that little area in red all the way to the right border of the watershed is the wetland, the natural wetland that I focused on for my project, and that is the Walnut Creek wetland, also known as Walnut Creek Wetland Park. It was designated as a park in 2009 and has since been a site of very notable urban wetland restoration. That actually started in the mid to late 90s by a neighborhood environmental justice movement. They were experiencing a lot of industrial dumping and just lack of purification of water that the city was not performing. So they formed a coalition environmental justice movement and had this park designated. And since then, they've had so many more resources from the city and volunteer work to help work towards restoring the wetland. So just as a disclaimer, I do work for this wetland. So if I say we, it is the royal we, and that I am referring to the park as a body, or myself and my coworkers. So we don't have very many formal monitoring processes at the moment. The city on a municipal scale is monitoring invasive plants such as Chinese and Japanese privet. Lesser celandine was an issue earlier this year. And we are also a site for the Wildlife Resources Commission's Box Turtle Connection project. But other than that, we don't have very formal monitoring protocols. And I wanted to kind of start building a foundation to change that. So for this project, I wanted to compile historical and current data that we had on mammal presence in particular, to create a comprehensive timeline of mammal sightings for the Walnut Creek wetland area, and establish a baseline mammal species list for the park that reflects recorded sightings. Now I've focused on mammals for a number of reasons. One of those reasons is not their status as indicator species for wetland health, especially considering the more subtle improvements that are being made in recent years and years to come. But rather, mammals are the most visible in terms of percentage of total species richness. And they're also the taxa that we have the most historical data for, them being the most visible. And this is also only in reference to recorded sightings that we have. I did not want to go through the complicated and arduous process of trying to record incidental sightings for the past 40 years. So that'll probably be a future project. So for this year's active data collection, for this year's timeline data, I used just regular camera traps. I had 5 different sites in a number of different kind of habitat types between unfettered wetland, and right off of State Street to the east. So pretty close to a lot of human activity. I have 5 separate sites, 30 days each, give or take, due to some technical difficulties. And then for our historical data review, we have a lot of past inventories in our archives. All of the individuals, except for the earliest one that we have, are people who have worked or continue to work very closely with the park. They are accredited or work with NC State. So it is reliable data. And then we also have archived trail camera footage starting in 2017. And that's where our official photographic evidence starts beyond the semi-published inventories. And as I mentioned, our first inventory that we have access to was done by the NC Geological Survey, just doing a general environmental assessment of the Walnut Creek area. None of us personally know the man who did this, but it was a government published document. So it was a great jumping off point. So this timeline has been split into 4 separate slides for legibility. But all of the years that we had data for are outlined in black, and each species has been marked the first year that they were recorded, and then each subsequent sighting in a colored dot. We have 20 total species through all of our records, and these are organized chronologically by the year they were first sighted. The common raccoon was the only animal or the only mammal species that we had that was recorded every single year of the data that we had. And then in kind of our first quarter, we have a lot more common species that you'd expect: white-tailed deer, beaver, which is a common wetland species, gray fox, possum. Oops, my arrow key is stuck. That's cool. We have some species that we only had recorded for 1 year, such as the tricolored bat, which isn't to say we don't have them or they didn't exist beforehand. I just wanted to have the consistency of physical recorded data. And then we're getting into our more uncommon species. We have a couple of species that haven't been seen in a few years, such as the mink. We do have photographic evidence of that, but we haven't gotten it since 2017. Actually, it might be 2018. That might be a mistake. But yeah, getting into our more uncommon species. And then 2017 was when we started having our personal, extensive trail cam footage. The northern river otter is not very uncommon in the park, actually, but it hadn't been recorded until 2017, and we've captured it every year since then. And then we also have some of our rarest species, such as the bobcat, which we only captured in 2018. And then our long-tailed weasel, which is very rare in the Piedmont at this point, which we only captured in 2020. And then we've been able to kind of start monitoring invasive mammal species, such as the feral cat and the nutria that had not been cited beforehand, at least in terms of an official capacity. And so it would be remiss to have a trail cam footage based project and not have any trail cam footage. So here are 2 primary invasive mammal species. We have our feral cats. We know we have at least 3. These are 3 separate ones from both last year, September 2020, and this year, September 2021. And then we have our nutria sightings, which I'm personally super jazzed about because I've never seen one personally. And the bottom 2 were my footage, and I have turned them into GIFs to view their behavior. And we know that they're not beavers because they have a long rat-like tail, and we know they're not muskrats because they're a little bit bigger. those. And then here's some of our rare sightings. These are animals that have not been sighted since these images. So they have potential to become species of interest as we develop our monitoring methods. We have our red fox there on the left in February 2019. This is our only photographic evidence of that. It was actually captured by Dr. George Hess's class, NR 300. He's in the Forestry and Environmental Resources Department. They've done a couple of full inventories of the park. So thank you to him for that. We have our bobcat sighting from January 2018. Excuse me. I am very interested in the lynx genus, so that is definitely on my further research, species of interest. And then we have our two rare mustelids, the mink on the left, which was last captured in January 2018, and then possibly our most exciting spotting that, as far as we know, no one else has ever seen, period, the long-tailed weasel, which we can tell is such because it is a little bit smaller, skinnier than the mink, and it also has a black tipped tail. It has also been corroborated with the Resources Commission, so it's legit. And then here is a hopefully working rolling mammal species list based on all of the recorded sightings we have. And then the right column is potential species to be further investigated, potentially added to the recorded sightings once we do have photographic or video evidence of it. So these incidental sightings are kind of examples, but they are examples that I have seen in the past year. And then possible sightings based on historic ranges. I do want to end up publishing this kind of list, at least the confirmed sightings for public availability, because we don't have anything of that sort for the park for any kind of taxa. And I think it's just useful information to be publicly available, not only for patrons, but for people who might want to conduct further research on the species that we have. And so that's part of my further investigation list to identify the species of interest that have only been recorded once or twice or not in a long time, and then develop more specific protocol and methods both on those species and having more sophisticated and consistent methods going to the future, and then creating more accessible records, as I mentioned, for public availability, and also for our use for both the staff in the city of Raleigh, as this is a very unique habitat. And we should be able to develop further records for more taxa, just to have this comprehensive inventory of all of our animals and plants. So thank you, special thanks for this research to my mentor, Liani Yerka, who has been constantly encouraging me and is so knowledgeable about the park and in general. And then Dr. Erin McKinney, who has given me their advice and support over a few years now in trying to work towards this minor. As we look at this very cute GIF of a possum scenting an Asiatic dayflower, do we have any questions?
Thank you so much, Adrian. I'm keeping an eye on the Q&A in the chat boxes.
Yes, Michelle, this possum is very adorable. I wish I could have included a few more just like behavioral clips, but they weren't super relevant. We got a lot of cute I admonish myself for calling this cute, but the nutria grooming itself, because ethically I can't call any invasive species cute, but it's just seeing its little hands clean itself is very good.
Well, in the interest of time, I'm going to propose that we move on to our next presenter, Shannon. But thank you again, Adrian. And anyone whose fingers maybe didn't move quite fast enough, please feel free to use the Q&A box. And Adrian, if you're able to stick around for a while, you can answer via text in the Q&A. So thanks again.
Mm-hmm.
Good job. And next, we'll yield the virtual stage to Shannon Dolan.
Awesome, thank you. All right, everyone can see my slides.
Perfect, good to go.
Great, thank you. Okay, hi everyone, I'm Shannon Dolan. I'm a senior majoring in environmental sciences, and I'm minoring in marine science, applied ecology, and technical and scientific communication. Today, I'm going to be speaking about the research I did during my second internship as a Dorothy Conservation Scholar, working with NOAA, the National Oceanic and Atmospheric Administration. My project was the effect of biological sampling effort on the precision of fish biomass estimates. And this was an analysis using R and NOAA's 2019 acoustic trawl survey data. So what is the acoustic trawl survey? Broadly, acoustic data is collected during the day and combined with the nighttime trawl data to generate species biomass estimates and stock distributions. These estimates are used by NOAA to inform fisheries policy. In this case, the acoustic trawl survey method is used to study coastal pelagic species, which are sardines, anchovies, herring, Jack and Pacific mackerel in the California current ecosystem. So daytime acoustic sampling involves an echo sounder, which is an instrument that transmits and hears sound waves. If the sound wave hits something, such as a fish or the sea floor, an echo is bounced back and recorded by the echo sounder. So now we have acoustic data that represents what we think is coastal pelagic species, but we don't know specifically what species it is. Is there anchovies or sardines? We don't know. And so that's where nighttime trawling comes into play. You go to areas where you believe coastal pelagic species echoes were recorded, and then you trawl. Usually 3 trawls occurred in one night and were summed into a trawl cluster. The proportion of species in the trawl helps us estimate how much acoustic data should be attributed to each species. So at this point, you may be asking yourself, why trawl at night and not as soon as you see what looks like coastal pelagic species backscatter? I asked the same question. And so the main motivation question for this project is, does trawling at night adequately represent species proportion? But this question is difficult to answer because we would need to trawl the same location in the daytime and at night and then compare the trawl catches. This would be time intensive and expensive. And so this project aims to address the sub-question, are biomass estimates and stock distributions sensitive to biological sampling effort, which helps answer if we are trawling enough to adequately represent species proportions, but doesn't really include that temporal variable yet. So the objective is to assess the assumption that trawling effort adequately and precisely represent the true species abundance and distribution. My hypothesis is that reducing trawling effort significantly decreases the precision of coastal pelagic species biomass estimates. And to study this, I wrote an R script to randomly remove a varying proportion of trawl clusters and recalculate biomass estimates and stock distributions. I had 3 different test conditions at 50%, 75%, and 90% trawling effort. The percentages are in terms of what amount of effort is being retained. For example, 75% trawling effort means I kept 75% of the trawl clusters and randomly removed 25% of them. And for each test condition, I generated 500 random samples each. And then to visualize and analyze the data, I created box plots, maps, and line graphs. Okay, so to illustrate part of what the code is doing, we're going to click through this animation. First, this is a map of the 2019 acoustic trawl survey area. The gray lines are the acoustic transects, which are split into 100-meter intervals for analysis, and then the black dots are the trawl clusters. So to estimate biomass, the closest trawl cluster to each acoustic interval needs to be determined. One way to do this is to run a line of code that creates polygons by radially expanding out from each trawl cluster. So all the acoustic intervals shaded in red would be apportioned to that trawl cluster. Now let's say we retain 90% of the trawl clusters. In this one random sample out of 500, that specific trawl cluster was removed. So now that original area shaded in red is split between the next two closest trawl clusters. You can see how much of a difference there is in area coverage. A similar thing occurs with 75% of the trawl clusters retained. Now with 50% of the trawl clusters removed, you can see how one remaining trawl cluster accounts for the same area that originally 5 clusters did. So my prediction is that areas with lower trawl species diversity will be less affected by reducing trawling effort. This is a map of the survey area where each pie graph represents the trawl cluster's acoustic species proportion. When comparing these two areas highlighted in the yellow boxes, if a trawl cluster is removed in box 2, the next nearest trawl cluster will also be majority anchovy, and thus the biomass estimate likely won't change that much. Whereas in box 1, if a trawl cluster is removed, the next closest trawl cluster has different acoustic species proportions and thus could result in a different biomass estimate. So let's take this a step further and look at a specific example. In this one random sample where 90% sampling effort was retained, 5 trawl clusters were removed, the ones with the yellow X's on them. This graph is sardines, where biomass is on the y-axis per species length on the x-axis. The yellow line is the biomass estimate using all of the trawl clusters, and the line in purple is the random sample. The sardine biomass estimate peak increased at lengths 24 to 27 centimeters because the trawl cluster that was removed off the coast of mid-Oregon was next to a trawl cluster with a higher proportion of sardines in the trawl catch. So the trawl cluster with more sardines is apportioned to more transects, hence a higher biomass estimate. Okay, so now we're going to jump into some overall results. On this slide, we have a box plot for Pacific herring, where the y-axis is the biomass and the x-axis is percent effort. The numbers in the bottom right-hand corner are for each stratum, which are drawn around contiguous areas where acoustic data was apportioned to the species. The dashed line represents the biomass estimate using all of the trawl clusters. The main takeaway here is that while there might be more variability and a larger interquartile range of potential biomass estimates with decreasing effort retained, the median biomass estimate at each percent effort still closely resembles the biomass estimate using all the trawl clusters, the dotted line. Okay, so now we're looking at sardines and Pacific herring, and the x-axis is fish length and the y-axis is mean biomass. Each line represents a percent effort test condition where all 500 samples are averaged together to get a mean biomass at each length. The first main takeaway here is that the peaks occur at the same fish length, and deviation from the biomass estimate using all the trawls, the yellow line, increases as percent effort retained decreases. The second takeaway is that some fish age classes are over or underestimated. For example, sardines length 15 to 18 are overestimated for 50% trawling effort, which might impact fisheries policy if you believe you have a large recruitment class aging into the fishery. All right, switching gears to look at area, we're looking specifically at jack mackerel and anchovy. The x-axis is percent effort, and the y-axis is the total stratum area for each species. The dotted line is the total stratum area estimated using all of the trawl clusters. The main takeaway here is there is a relatively small change in total area, even at 50% effort retained. And as the percent effort retained decreases, the box plot median area deviates further from the total area estimated generated with all of the trawl clusters. Okay, so now let's look at the geographic distribution of sardine stratum. This is the map of the survey area where the bold black box is the stratum area, areas generated using all of the trawl clusters, and the colors represent each stratum. The original sardine data did not include a fourth stratum, but occasionally and increasingly with a reduction in trawl effort, a fourth stratum was generated. Also, the geographic range for each stratum extends further and overlaps with other stratums at 50% trawling effort. The main takeaway here is that reducing trawling effort does result in more variation in stratum geographic location. So now revisiting my hypothesis, which is reducing trawling effort significantly decreases the precision of coastal pelagic species biomass estimates, my hypothesis is somewhat supported. Right, we saw a minimal difference in the total stratum area, and the biomass estimates had peaks at similar fish lengths, but it's context dependent. It really depends on what are the acoustic species proportions of the next closest trawl cluster when you remove one. And so the results support that the current amount of trawling is robust. And the most compelling graph to support this is the total biomass box plot, where the median biomass estimate doesn't deviate much even with a reduction in trawling effort. Regardless, I would not recommend trawling less because in real time on the vessel, you might, like how do you determine where you might be able to trawl less? You could consider historical trends, but climate change is altering our ocean system, so historical trends might not represent current conditions further or anymore. And then for future research, I would go back to my main motivation question and address the temporal differences by trawling the same area at night and during the day and generating biomass estimates from those. So a huge thank you to my two advisors at NOAA, Dr. Steerhall for his coding expertise and Dr. Deemer for sharing some of his extensive fisheries acoustics knowledge with me, as well as a thank you to Dr. Leggett and Dr. Rena for their support through the Dorsey Conservation Scholars Program. So with all of that, thank you. Are there any questions? Okay, so I see one in the chat. Validating the identity of acoustic targets is always challenging, absolutely. Given that many species exhibit dielphitic migration, how confident are you that the organisms detected in the daytime acoustic survey are actually the same as those collected in nighttime trawls? That is a great question and part of what this is trying to answer. You know, it is difficult with that temporal variation that, you know, you're going back and surveying at night, but how much are fish really moving during the day? And then also part of why we trawl at night is that it's difficult to trawl and collect coastal pelagic species in the trawls during the day because they are schooling and they're able to avoid the net. Whereas at nighttime when it's darker, they have less visibility, they can't see the net, and so they're able to be actually captured. So there was some difficulty in like real world that we couldn't actually trawl during the day. Also another question, did a lot of data analysis. Do you have much experience with data analysis support? No, I didn't. I actually downloaded RStudio a couple months before I started this project. And then with the help of Dr. Steerhall and using some of his code, I was able to write over 1,000 lines of code to do this analysis. I really learned a lot about how to create graphs and visualize data and, you know, do fisheries work with R, which is really awesome and a great skill set to have now.
Awesome. Thank you so much, Shannon. And thanks also for that plug for RStudio and building your professional development skills, which I think a lot of these minor research projects do. Up next, we have Malia Naumchik. Malia, are you ready to go?
Yes. Okay. Ooh.
Yeah, that's great.
You can see my screen?
Yep, perfectly.
Awesome. Let me move this. Okay. All right. All right, so hi everyone, I'm Malia Naumchik, and I'm going to be talking about a study I conducted this semester, investigating whether the weight of pollen a bee is carrying affects their body temperatures. Okay, so first I'll start off with a little bit of information on the bee that I used in my study. I looked at worker bees within the species Bombus impatiens, also known as the common eastern bumblebee. This is a very widespread species of bumblebee found through most of the eastern half of the United States and parts of Canada. And I chose this bee because it's abundantly found in Raleigh. It has a long flight season well into the fall when I was collecting my data, and it's a really important native pollinator that's commercially used in agriculture across the country. And now for some background on why I conducted this experiment. We're experiencing widespread declines of pollinators across the world due to climate change, and bees are particularly vulnerable. Bees, especially bumblebees, due to their large size and furry body, can generate a lot of heat while they're flying, which can make them prone to overheating at high temperatures. So as climate change continues, we might expect to see more drastic decreases in bumblebee populations. Also, pollen foraging might make bees even more susceptible to overheating. One study found that pollen foraging honeybees had higher metabolic rates than nectar foragers, and another study showed that pollen foraging stingless bees had reduced flight durations than nectar foragers at high temperatures. So using this information, I wanted to look into how pollen foraging affects the body temperatures of bumblebees. And if bees carrying large amount of pollen are hotter, they might be pushed towards their upper thermal limits. And this limit is commonly measured as the critical thermal maximum, or the temperature that the bee loses basic muscle control and function, which is ultimately lethal. For bombus impatiens, a previous study in my lab found that their range of CT max temperatures was 42.6 to 49 degrees Celsius. And I'll talk about why that's important later in my presentation. So my two basic research questions with this study were, are bees carrying a large amount of pollen hotter than those carrying a small amount of pollen? And if they are, would this increase in temperature push them within the range of their CT max? And my prediction for this study was that the body temperatures of bombus impatiens would increase as the weight of pollen they are carrying increases. So to test these research questions, I went to the J.C. Ralston Arboretum in Raleigh, North Carolina, and I collected 96 bee and pollen samples from early August through late September. And I collected those samples in the Rose Garden and Color Trials Garden, which you can see on the aerial imagery on the right. And to collect the temperatures of these bees, I quickly captured them off a flower into what is called a bee squeezer, which you can see on the left. It consists of a foam plunger that is used to push the bee up within a small cup that has fine mesh on the top. In this, I flipped the bee onto its back, and within 1 to 2 seconds, I inserted a thermocouple probe into its thorax to get its internal thoracic temperature. I then anesthetized the bee using CO2 and removed the pollen from its hind legs using a brush pick while it was unconscious. After that, I transferred both the bee and pollen into separate vials that I later weighed in the lab using a balance. And because I did this study in the field, I had to have a way to account for the environmental effect on the bee's body temperature. To do this, I used an operative body temperature model, which has been used in several studies on bees and other insects. For this model, I used a dead bee placed in a lifelike position on a flower near where I was collecting my bee samples. It was attached to a thermocouple that recorded temperature readings every 2 minutes. And these readings from this model served as a baseline of what the bee's temperature would be based on their environment. I could compare the live bees' temperatures to the operative body temperature measurements, and any variation I saw would be due to their own metabolism and heat generation. And once my temperatures and weights were measured, I performed a multiple linear regression, looking at how body temperature was correlated with operative temperature, body weight, and pollen weight. And I included body weight in the analysis because large bees are usually hotter than smaller bees. Bombus impatiens workers have a large variation in sizes. I had bees ranging in size from 70 milligrams all the way up to 300 milligrams. So I wanted to account for as many variables as possible. So here are the results of that multiple linear regression. The first graph on top represents the relationship between operative temperature and body temperature when holding bee weight and pollen weight constant. There was a significant relationship, and this was expected as bees do depend on their environment as a major heat source. And next, I did see a significant relationship between the bee's weight and body temperature when holding the other variables constant, which was also expected based on previous studies. And finally, I did see a significant relationship between pollen weight and body temperature after accounting for the other two variables. So this result did confirm my prediction that bees carrying a large amount of pollen are hotter than those carrying only a small amount of pollen. There was actually about a 0.07 degree increase in body temperature for every milligram of pollen added. And so now that we know that bees with more pollen do tend to be hotter, would this put them at risk of reaching the critical thermal maximum? I created this chart of the range of CT max values measured for bombus impatiens from the previous study in my lab, and what the theoretical temperatures of large bees and small bees would be based on whether or not they are carrying a pollen on a hot day. So a small bee carrying no pollen would only be about 41 degrees and wouldn't really be at risk of reaching their CT max. But a small bee carrying a full pollen load could reach a temperature of about 42.7, which puts them just within the range of temperatures that could be lethal for them. A large bee carrying no pollen on a really hot day would be about 43 degrees, which puts them within the range of their CT max, but might not be lethal for all bees. And finally, a large bumblebee carrying a full pollen load would reach a temperature of about 46 degrees, which puts them right at that average critical thermal maximum measured for this species. So in the future, as we have more really hot days, bees carrying full pollen loads are going to be more at risk for reaching their CT max, with larger bees being especially vulnerable. And this is really worrisome because bees might reduce their foraging trips, which would negatively affect their ability to pollinate crops and would decrease the amount of pollen that they could collect for their larval provisions. And I'll briefly talk about a few minor pitfalls that I encountered during my research. First off, the operative body temperature model I used was hard to position on the flower sometimes because it was really heavy. To fix this, I could use an adhesive to fix the bee onto the flower, and maybe using an infrared camera to continuously measure the temperature so I wouldn't have to position the thermocouple. I also did have some unexplained variance within my data. Because this was a field experiment, there definitely could have been more variables that I couldn't account for. So to solidify my results, I could perform a controlled lab experiment in the future. So why was my study important? Previous studies have looked at the increase in metabolic rates, temperatures, and mechanical power outputs with increasing load weight, but most of those studies have been done on honeybees in the lab. Only one study looked at the effect of added weight on body temperature in bumblebees, but it only looked at the nectar crop load, and it was also done in the lab. So my study was the first field experiment to demonstrate this correlation between load weight and body temperature. My results indicated that pollen foraging bees could be negatively affected at high temperatures. So as temperatures continue to warm due to climate change, bees' pollination services to farms and native flora might be at risk. So it's essential that we have more research on this topic. So what would future research look like? I would like to look at how this phenomenon affects worker behavior in the field. At high temperatures, would bees start to seek shade, bring back smaller pollen loads, or even stop foraging? Also, would high temperatures affect the total amount of pollen brought back to the nest? Pollen is essential for feeding their larvae, so decreased pollen could lead to decreased colony size. I would also suggest looking at other species, including Bombus pennsylvanicus, which you can see on the right. This bumblebee has already seen declines in their population, and it's usually larger than Bombus impatiens, so it might be more at risk. And I'd also like to see if there are ways that we can help bees cool down based on this research. If pollen foragers are at risk for overheating, would providing them with bee baths or shade help them stay cool? And finally, I'd like to thank my mentor, Dr. Elsa Youngstedt, for helping support me during this project, and Douglas Ruin, the gardens manager at the Arboretum, who allowed me to conduct my research at the gardens. And now if anybody has any questions, feel free to ask them or put them in the Q&A. Thank you.
Thanks, Malia. You have a question already in the Q&A box.
Do you think that your temperature measurements would have differed if you used an anesthetized bees instead of a dead one? Are you referring to the operative body temperature model, or? Yes, okay. So if I had used an anesthetized bee, that's hard to answer, because the dead bee basically would be what the temperature would be without metabolism. So that was helping me see the variation in temperature due to their own heat generation, if them carrying pollen made them more hotter. So I think that if I use an anesthetized bee, it wouldn't really give me the same kind of information, I guess, if that answers your question.
I guess a follow-up, was it a freshly dead bee?
So I had to dry it some in order to get it to stay in its position. I had to leave it out usually for like 1 or 2 days, and then I would do my research on the weekend, and then after that I would throw it out and then do another one. So it was about 1 or 2 days old when I used it.
Not to beat a dead bee, but. And you have a question, a shout out in the chat from Michelle Kirchner for your original artwork. I didn't realize that was yours.
Yes, I made that.
Very well done indeed. That's gorgeous.
Okay, I can go ahead and stop sharing my slides then.
Well, thank you so much, Malia. Up next, we have Kimmy Gebbia.
Okay. Is everything up there working?
We can see and hear you.
Okay, perfect. Well, good afternoon, everyone. My name is Kimmy Gebbia, and today I will be discussing my research project on urban development and Falls Lake water quality changes over the past decade. Oh, okay. So across the United States, more than half of the rivers are already too polluted for human health and for the health of aquatic life. So stream health nationally has been declining, and it has been linked to increased urbanization and stormwater runoff pollutants. And Raleigh is no exception to this trend. Wade County has grown by 43.5% in the past decade and is gaining 100,000 people every year, according to the US Census Bureau. Raleigh is the second fastest growing metro behind Austin, Texas, and Raleigh and Durham have grown 23 and 26% respectively in the past decade. So these increases in population are not only leading to more watershed development, but they're also placing higher demands on Falls Lake, our drinking water supply. So Falls Lake is located about 40 minutes north of downtown Raleigh and is the drinking water source for Raleigh and 6 other municipalities. In total, it supplies water for about 600,000 people, and that continues to increase. It also already has incoming streams and parts of the lake that are impaired. So if you'll see on the map in red, everything that's highlighted in red, they're already violating water quality standards. And these are standards set by the state such as by too much algae indicated by chlorophyll A and other nutrients such as turbidity and total suspended solids. So I use GIS as part of my project to look at impervious cover and land use changes. This map is displaying the impervious cover in 2008. The lake here, we chose to split it up between the upper and the lower reservoir. So this was mainly because the lower reservoir at the bottom at the end has the water intake to our drinking water supply. And so we wanted to split it up and look at them differently. And it's also worthy to note that 2/3 of the stream inputs go into the upper lake. So there's a lot of nutrients. A lot of those streams were impaired, if you saw from the previous map. And then we have Durham as a major influence on the upper watershed, and Raleigh has a lot of encroaching suburban on the lower watershed. And so if you'll pay attention to the warm colors, as I switch to the next slide, it'll show the same map, but for 2019, and you can see where impervious cover has increased. So I'll switch back and forth a few times. So it doesn't look like there's a ton of change here in the impervious cover, but a little bit of impervious cover change has a powerful impact on the water quality. So research has shown that greater than 2 to 5% of impervious cover affects, it degrades surface waters and affects biota. So these small increases in impervious cover are actually heavily affecting the reservoir. So from these GIS maps, I was also able to determine that there was a 5 to 6% increase in developed land use from 2008 to 2019. And in 2019, approximately 5% of the watershed was classified as impervious cover. So that meets that threshold of 2 to 5% impervious cover degrading surface waters. So for my research question, I looked at how has urban development influenced water quality in the upper versus lower lake over the past decade. And I expected to see water quality degradation lake-wide with a much stronger correlation in the upper lake, again, due to those 2/3 stream inputs that are already degraded and already violating water quality standards in the upper lake. And then I also expected to see strong correlations with algal biomass in development, and algal biomass was indicated by chlorophyll A. And then I also expected to see strong correlations with the major algal nutrients, total nitrogen and total phosphorus. So in order to assess these predictions, I first determined how land cover had changed over the 11-year period. I obtained national land cover data for every 3 years and analyzed it via GIS. We had 5 raster data sets, and they were each extracted and clipped to the Falls watershed, as indicated in the image on the slide. And then the area and percent change for each of the 15 land cover classifications were then calculated. The land cover was correlated with water quality data. So I was given a water quality set from the Center for Applied Aquatic Ecology. They have been collecting water quality for years on Falls Lake. And so I looked at the water quality data from 2008 to 2019, and that was about 185,000 data points. In each of these parameters were analyzed based on the upper region, the lower region, reservoir wide, annually, and seasonally. But for this presentation, I had to simplify it down. So I ended up focusing on just 3 major variables, which was chlorophyll A, nitrogen, and phosphorus, and then only looking at total developed land split up between the upper lake and the lower lake. I then graphed scatter plots and looked at the Pearson correlations. So a Pearson correlation coefficient is a measure of the strength of a linear association between 2 variables, and greater than 0.7 is a strong correlation, and then 0.5 to 0.7 is a moderate correlation. So this slide shows algal biomass as chlorophyll A versus the total percent change in the urban watershed. The first graph on the left is the upper lake and the upper watershed. There's a lot of scatter and chlorophyll concentrations, and they're all pretty high levels. And notice the last line of dots, the red indicates the overall change for the 11-year period, which was a little bit more than a 4% increase in urban development in the upper watershed. And then we have the lower lake. The lower lake had more development, a higher development increase, almost 13%, but it started from a much lower amount of development. So there was a positive increase, a positive correlation in the upper lake between chlorophyll and urban development, and then no correlation, no change in algal biomass in the lower lake. And this graph shows total nitrogen for the upper lake. It's total nitrogen versus the change in urban development in the upper watershed. And there are, again, high nitrogen concentrations, indicating a lot of incoming pollution throughout the study period. But there was no correlation between TN levels and further changes in the urban development. But we suspect that this is because Durham has had a lot of improved sewage treatment, which likely offset the small but potent increase in urban development temporarily. And then in the lower lake, we see a lot lower values of total nitrogen concentration. And then there was also a negative correlation between total nitrogen and the change in development, but I will show the rest of the data and come back to that. So again, the same graph set up for total phosphorus. We have total phosphorus and the change in urban development. TP was very high in the upper lake throughout the study, and there was a strong positive correlation between increase in watershed development and TP concentrations. And then again, for the lower lake was generally lower concentrations for TP, and then again, a negative correlation between TP and development. So the data show in general that the upper lake had much poorer water quality throughout the study, and increasing pollutants TN and TP were strongly correlated to increased urban development. But what is going on in the lower lake? So the lower lake, we suspect, has strong dilution. The low levels of pollutant in general already have been substantially diluted due to 2 very, very wet years at the end of the study in 2018 and 2019. And then we looked for evidence of these wet periods, and this was supported by additional analyses. So we took off the final 2018 year and looked at the correlations. And once we did that, nitrate was positively correlated with the development in the lower lake. And then chlorophyll and TP were also moderately to strongly correlated between 0.6 and 0.7 with low, medium, and high density in the lower lake, specifically when we also only looked at the summertime. So these preliminary findings suggest that the correlations were confounded by climate variability and change. Another possible influence were septic systems. So this map indicates the amounts of septic systems in 1990, and there are definitely a lot in the lower lake. But over the past decade, we've seen a lot of these old leaky septic systems being replaced by new houses and new improved septic systems. So this is possibly creating a temporary decrease in concentrations of pollutants entering the lower reservoir. However, with time, it's expected that these concentrations will increase again. So in summary, Falls Lake is rapidly urbanizing, with Raleigh as the second fastest growing metropolitan area. And there's already parts of Falls Lake that are surpassing the 2 to 5% impervious cover, degrading surface waters, and there's already impaired waters. So for my study, I analyzed watershed land cover data and tested for correlations, and we found that the upper lake, algal biomass and TP were positively correlated with increased urban development, while in the lower lake, algal biomass didn't seem to have any effect, and then N and TP were negatively correlated. But once we took off 2019, because of the very wet years, we found positive correlations again. So overall, the data suggests that development is degrading the water quality, not as much in the lower lake, but the lower lake doesn't seem far behind the upper lake. And there's just not really a lot of control over development, resulting in a lot of difficulty protecting our drinking water source. And this is an issue across the country. So if this is a situation of concern to you, there are a few suggestions that we've put together that we would suggest. So just find out more, have a strong education, have a strong educational background on what's going on and be able to talk about it. Also paying attention to politics, check voting records, keep up with legislation, call your representatives if there's something going on that you want to be voted for or against, and to join in on communities and organizations that are fighting for our natural resources. So thank you for your attention. And I'd also like to give a special shout out to Ellie Allen, who is my mentor through this whole project and guided me through it this whole semester, and also to Carol Kinder for her expertise in data and statistics, and Dr. Joanne Burkholder for her expertise in Falls Lake Reservoir and understanding what's going on and all the ecological concepts behind what's happening in the lake. So, any questions?
Thanks so much, Kimmy. We have a question already in the Q&A box from Jim Rice.
Does the development happening not abide by the current? I think the current regulations are not adequate. I think a lot of the time data is misused or skewed so that the trends are ignored, and developers just kind of are, the policies are increased so that more development can happen rather than being held accountable for all the things that are going on. So yeah, I think they're just not adequate.
Well, thanks again. And once again, an open invitation to anyone who has questions as we go. Feel free to continue to type those in. Let me check the chat room. All right. So up next, we have Davis Grubb presenting.
Let me just go ahead and share my screen. Hello, my name is Davis Grubb, senior studying environmental science, and my research project I worked with was on assessing blue crab nursery habitat. So first off, what is exactly nursery habitat? Well, nursery habitat is any habitat that juveniles of a species use to, well, grow or spend basically their childhood there. These habitats protect them from predators, they provide food, and they support population persistence, as in new juveniles can enter after they spawn in. So blue crabs have a very complex and interesting life cycle. As you can see, they begin life as basically microorganisms in the middle of the open ocean, eventually making their way into the coastal areas of the US, where they'll settle into nursery habitat as first instars, which is basically when they start actually looking like, well, crabs. And they usually will settle into either two types of habitat at seagrass habitat or SDH or sallow-detrital habitat, which we were interested in looking at. So why are blue crabs important and why are we studying them? Well, blue crabs they provide sustenance for us. They're a food source we eat. They also play an important role in ecosystems. They're food for other animals. They're omnivores. They consume plants and other animals. But one of the big things that we're concerned with about the blue crab is the blue crab has actually been in decline since the late 90s. And we think that might be because their habitat might be deteriorating, specifically their nursery habitat. So what is shallow habitat and why is it important? Well, it's an SDH, or is a soil type which is found in and around marshlands and is one of the two main nursery habitats for blue crabs. And our research objective was to look at the variabilities of soil samples from SDH. So what I was doing is before I joined this project, a number of known SDH samples were collected and analyzed. So what I am looking at is actually several site surveys from along the coast of North Carolina to see what soil is and is not SDH. So our methods. So as I said, 2019, wherever you can see an orange dot, a core was taken, and what I did was I analyzed it. Cores, as I said, were also taken from known locations, and they were analyzed previously. So what did I do to analyze them? We took three different things into consideration when analyzing the soil. We took in water weight, combustion, and grain size. I did not actually do the water weight analysis, but I did combustion and grain size analysis. And basically to do these, for example, to do combustion analysis, we would take a sample and we would run it through a kiln where all the organic matter would be burned off. So when we compare the weight before we burned the sample and the weight after, we could see the percentage of organic matter in the sample. And for the grain size analysis, it's much easier to explain. We basically ran the sample through a bunch of pans, as you can see here to collect all the particles, and then we weighed all the collected residue to determine what percentage of the sample was what grain size. And so the previous results for what were the known SDH cores was SDH has a high water weight, high organic matter, and a high proportion of three different grain sizes. 4.5 millimeter grain size, which is large particles. SCH is made out of deteriorating marsh grass. So this is mostly things like roots of the marsh grass. 400 micrometers, which is the peat mat, which is basically some of the more deteriorated parts of the marsh. And then 63 micrometers, which is that fine organic matter as the marsh is basically being ground up. So this first graph showcases our the percentage of the water weight we found. And as you can see, it was a bimodal distribution of the cores I sampled with a good amount being around this 20% mark, and the other amount being around this 80% mark. Now, known SDH is usually going to be in this orange graph. As you can see, the blue graph to the side is what was found previously among all the SDH cores. And so the corresponding, the cores that I looked at that would be SDH would be in this orange graph here. We did the same thing with organic matter. Organic matter also had a bimodal distribution with a lot of samples being in around 0% organic matter. These were sandy soils. These were things like beach or gravel, sand. And while the second distribution was around this 20 to 30% mark. Now, SDH, the known SDH had actually a very wide variability of organic matter between 20 and 70%, which majority of our samples that I looked at fit in. Now, where there was some discrepancy was in the grain size. So we looked at 6 different grain sizes, actually, not pictured here, including 1.2 millimeter and 250 and 150 micrometer, because we did not find really enough of it to really map out what SDH is. But as you can see from the core samples that I looked at, a lot of them were actually fairly low in the 4.5 millimeter and the 400 millimeter here, which means a lot of the samples that I looked at for their grain size analysis do not appear to be SDH, although most of our samples did fit into this 63 micrometer grain size. But that is probably because most soil types will have very fine organic matters like mud and clay and all that. So conclusion, so as I said, water weight had a bimodal distribution. Some samples had very low, very high water weight, and with high water weight being indicative of SDH. For organic matter, we again had a bimodal distribution between samples that had fairly high organic matter to samples that had almost no organic matter. And our grain size sample, we actually had a very low 4.5 millimeter, 400 micrometers, but most of our samples had a very high proportion of 63 micrometers in grain size. So moving forward, this project is still ongoing. Aaron Voigt, who's a leader of this project, actually collected some more cores to evaluate this over this summer. And we're going to do a logistic regression samples to compare some more of them to our known SDH samples. So yeah, I'd like to thank Aaron Voigt, who's my project manager and mentor for this. I'd also like to thank Ecosystem Lab, the North Carolina Sea Grant, and the AP and EP for all making this project possible. I'd also like to thank Aaron McKinney for helping me find this position and helping me introduce to Aaron Voigt in the first place. So yeah, any questions?
Thank you so much, Davis. I've got the Q&A and chat open. So between you and I, we will catch any questions that come through.
I know soil isn't exactly the most exciting subject for a lot of people.
No, I think we are experiencing the classic lag of thinking to typing. Michelle is a big fan of Soil's work.
That's good to see.
And in case folks are still gathering their thoughts, we can definitely keep an eye on the Q&A and you can answer any questions that come in via text. So I'll say thanks again. And if you don't mind, stop sharing your screen.
Okay.
We will wrap up the fall 2021 Applied Ecology Minor Research Symposium. Last, certainly not least, Corrine Morales.
Hello. Okay, I gotta share my screen. Is it showing up? I don't know if it is or not.
I don't see it yet.
Okay, well, is it there now?
Yes.
Okay, good. Sorry.
No, nice. Good to go.
Yeah. So my name is Corrine Morales. I would put on my video, but my Wi-Fi is not very stable, and I would like to actually be able for you guys to hear me. So I looked into longleaf pine stand characteristics in gopher tortoise habitat, or alternatively, the effect of longleaf pine spacing and canopy openness on legume cover in gopher tortoise habitat. As a disclaimer, this data is part of a larger project that I've been working on with a grad student, Jax Housley. So we've been collecting a lot more data than this and a lot of different sites. This is just from one specific site and with certain characteristics. So for background, the gopher tortoise is listed as a vulnerable species on the red list right now. It's also a keystone species that has burrows that provide shelter and protection to other species, especially when burning these stands, which is very important for the longleaf pine species. And there's a little toad over there, gopher frog, as one of the species that goes in the burrows. And the bulk of the gopher tortoise diet is made up of grasses and legumes, which is why I chose to look at legumes specifically. Longleaf pine only currently occupies less than 3% of its historical range because it was used for turpentine and it was actively overly harvested historically. So currently there are a lot of replanting efforts to restore its range. This is its original range, and like I said previously, it now occupies less than 3% of this. Furthermore, when planting longleaf pine or planting trees for gopher tortoise habitat, there are two different recommendations going on. The longleaf pine density is typically recommended at 545 trees per acre. That also varies anywhere. We sampled areas that had 500 to 800 throughout this whole project, but the typical was around 700 too. The pine basal area less than or equal to 70 square feet per acre for the gopher tortoise habitat, which is obviously a big discrepancy. The effects of restorative planting is also not completely known in legume growth and presence. And spacing may determine how much space is available for tortoises to browse the vegetation and the space needed for their burrows. And their burrows are upper in the right corner. I did see a lot of those in my internship. I only saw 2 gopher tortoises throughout my whole internship though, but happy I got to see at least one. And canopy cover may limit incoming sunlight that is available for these legumes to grow, which is why we looked into these characteristics. So the objective of this study, the overall parent project of this, like I said, with the grad student I've been working with, is funded by the NRCS, the Natural Resources Conservation Service, and they typically help private landowners plant longleaf pine and for gopher tortoise habitat, and they give them a forester and they set it up for them. So possibly this could help inform what would be better for the combination of longleaf and gopher tortoise. So from our first hypothesis, I hypothesized that as canopy openness increases, lagoon cover will also increase because of the increased sunlight. And the second hypothesis is as spacing increases, lagoon cover will also increase just because more growth. And our methods for vegetation, i.e. the legumes, we used a tool called a Wien's pole, which is shown in that picture. Ours looks slightly different, but it gets a message across. Ours had markings at 0.1, 0.2, 0.3, 0.4, 0.5, 1, 1.5, and 2 meters. And that was marking the height of each of the vegetation that we took, and measurements were taken in 4 directions, north, south, east, and west, and 5 steps in each direction, 1 in plot center. And we tried to be unbiased as possible, so we wouldn't be looking down to try to get certain things in our measurements, avoid bald spots. We try to be as unbiased as possible. And the lagoon data that we had, because we surveyed for other vegetation overall, as in other woody plants, vines, grasses, wiregrass, a bunch of other stuff, ferns. We pulled that lagoon data from that data for this analysis. We also took hemispherical photos for canopy cover, and these were taken on site. We made sure the camera was at 1 meter tall and was perfectly level. And we analyzed it using the gap light analyzer, which is the picture we would take in the stand is shown here. And then we would take the picture in the gap light analyzer, choose a color plane, then a threshold, and convert that to a black and white image. And then the percentage of white pixels over the total would give us the canopy openness. Spacing was measured with a 50-meter tape measure and rows and columns for the spacing of the plots in a 10-meter plot that were randomly selected. And then that is our math, which is the 43,560 divided by average column width times average column row width rather. And that number is the number of square feet in an acre. And furthermore, our methods, all these measurements were taken at the Jones Center at Itchaway in Newton, Georgia. These measurements took place from June 17th to the 22nd of 2021. We had a total of 47 plots and 10 stands in total, and approximately 4.7 plots per stand. And stands are just an area of pine trees that have similar characteristics because they undergo the same management. So that's what that means. All stands were previously burned on different intervals because that does affect lagoon growth. And our goal with this study overall as well was to have a maximum age of 8 to 10 years for our stands. And we ran a correlation test and a t-test. So these are the correlation results for spacing and lagoon cover. There was a negative correlation here, and we used the average for each stand rather than each individual plot. Same thing with canopy openness and lagoon cover, again, a negative correlation. Our t-test results showed a significant value. Both null hypotheses were rejected. And both canopy openness and spacing showed a significant negative correlation with lagoon cover. And that is a picture that I took in one of our stands. Furthermore, with previous research, people have found that gopher tortoises are more sensitive to shading rather than the availability of vegetation, say legumes. So with this data in mind, it might not be as negatively affected with a decrease in available legumes in open canopy because of this preference. There weren't very many pitfalls with the research itself. I mean, of course, we got our truck stuck in the mud once in the overall project, but there were minimal complications with the research itself. One minor one was for the hemispherical photos. There need to be minimal sunlight because then that would affect the way that the camera perceives the canopy. So we just adapted our strategy to take these pictures as early as possible to avoid this. But that did mean waking up at like 5 o'clock in the morning, but we did it. Also, as I said, how can this data be applied? The NRCS is actively advising private landowners, and private landowners can possibly use this information if it is public, probably with the larger parent project to this one to be more complete and overall, and have an informed new standard for planting in gopher tortoises and longleaf pine stands. And future research could include specifically just about grasses instead of legumes, since they, like I said before, legumes and grasses make up the bulk of their diet. You can see the gopher tortoise munching on some grass over there. And also the nesting behavior differences with differing spacing and vegetation cover, because they need the space to make their burrows, because they are fairly large, especially with the way that they dig, and vegetation cover because obviously these animals are low to the ground. So maneuverability is very important. And overall acknowledgements, obviously special thanks to the Jones Center for letting us visit and conduct our research there. It was a great start off to our experience. Also the NRCS for funding the parent project and supplying all of our site information and the mapping that we used. Also thanks to my faculty advisor, Dr. Jody Forrester, right there in the middle, and Jax Housley, my advising grad student. They have helped me so much, and that's us with our machetes in the field. So if there's any questions, feel free to let me know.
Thanks so much, Corrine. Final call and last chance to get all your questions out. Hearing none, and maybe giving some folks some time to type up questions if they still have them. A huge, huge thanks to all of our student presenters. This has been amazing. This is like a very joyful experience for me every semester. I'm never happier to sit in a 3-hour Zoom meeting than when it's in celebration and recognition of all of your hard work. It's really remarkable. I hope you all feel really incredibly proud of your accomplishments. Yeah, Michelle has unmuted video to clap, clap, clap. A huge thank you to all of our research mentors, faculty, grad students, postdocs, and those beyond academia who make this work possible. Thank you, thank you, thank you. If you want to join the network of folks who I email regularly, I will just drop my email in the chat. Drop me a line. Let me know that you want to work with undergrads. As you can see, our undergrads are spectacular. And huge thanks to all of you who have attended today to watch and support and hopefully learn more about the diverse and amazing research that the students in our minor program do. I know 3 hours is a long time to be on Zoom, but really, really grateful that all of you stuck with it right through to the end. And we've got, a lot of praise coming in on the chat. So thank you all so, so much. I hope the end of your semesters wrap up smoothly. I hope you all are able to take a much deserved and well-earned break over this next coming week for Thanksgiving and Native American Heritage Day. Congratulations all. Thank you so much.